My name is Kiman Lekau. I'm a professor in statistical genomics and director of Melbourne Integrative Genomics, and I'll be sharing this public lecture today. Before we start, I would like to acknowledge the traditional owners of the unceded land in which we work, learn, and live, the Wurundjeri Wurrung people of the Kulin Nation. We recognize the unique place held by Aboriginal and Torres Strait Islander peoples as the original owners and custodians of the lands and waterways across the Australian continent, with histories of continuous connection dating back more than 60,000 years. We also acknowledge their enduring cultural practices for caring for country. We pay respect to elders past, present, and future, and acknowledge the importance of Indigenous knowledge in the academy. As a community of researchers, teachers, professional staff, and students, we are privileged to work and learn every day with Indigenous colleagues and partners. So welcome everybody to this public lecture of BioInfoSummer 24. For those who just joined us now, BioInfoSummer is a national training and networking event where we bring together students, early career researchers, and professionals from mathematics, statistics, data science, biology, biomedicine to explore current bioinformatics research and developments. We would like to thank the sponsors without whom we wouldn't be able to have BioInfoSummer 24. So this is the Australian Mathematical Sciences Institute, AMSI, Australian Biocommons, the Centre of Excellence, MAXIS, Melbourne Integrative Genomics, and the School of Mathematics and Statistics at the University of Melbourne. It's my great pleasure to introduce you to Professor Daniel MacArthur. Professor MacArthur is the director of the Center for Population Genomics, established to solve critical scientific, regulatory, and technical problems that currently limit the development of genomic medicine in Australia. His center aims to execute large-scale projects that will establish Australia's leadership in this field. Professor MacArthur previously served as co-director of the Medical and Population Genetics program at the Broad Institute of MIT and Harvard. He co-directed the Broad Institute Center for Mendelian Genomics, which has sequenced and analyzed genomic data from over 10,000 individuals from rare disease families and contributed to the discovery of more than 100 new rare disease genes. He also led the development of the Genome Aggregation Database, which is the largest and most widely used dataset of human exome and genome sequence data. And they have collated so far more than 140,000 sequence individuals. So it's a great pleasure for me to, and for us to listen to your lecture, Daniel. You will give us an overview of what is the new field of genomic medicine and what it means for the future of health, and what Australia needs to do to ensure that all our citizens can benefit from these powerful new genomic technologies. Thank you, Daniel.
Thank you very much. Can you guys hear me okay in the back? That's good. Good to hear some nodding. So thanks very much for that kind introduction and for the chance to present here today. So I'm delighted to be here. Sounds like it's been a great meeting for those of you who've been at the meeting. I'm going to give an overview today that's very high level. So for those of you with already with a deep sort of biology or bioinformatics background, a lot of the introductory material will be very familiar. But I'm going to talk right towards the end very briefly about some of the work that my own team is doing. And then I'm really happy in the Q&A to go into more detail about any of the projects that we're working on that strike you as being of interest, or answer any other questions that you have. But I thought for today that I'd start with a family that I think illustrates really nicely why I'm so excited about the power of this new field of genomic medicine. This is a family, they're based in the US. They have 3 kids, all of them tragically affected by the same very severe genetic disorder. This is a disease called spinal muscular atrophy. It is classified as a rare genetic disease, but is actually one of the most common rare genetic diseases, particularly in individuals of European ancestry. It affects 1 in every 3,000 to 5,000 babies born in Australia each year, for instance. SMA is an awful disease. If you were born with this disease a decade ago, it was a death sentence. And it kills kids by paralyzing muscles throughout their body, eventually resulting in them not being able to breathe. And the 3 kids in this family, again, are all affected by the same disorder, but thanks to the development of genomic medicine, they've had very different outcomes. On the left-hand side here, you can see the oldest son, Matteo. Matteo was born before the advent of any effective therapy at all for spinal muscular atrophy. And although he did receive an early form of treatment at the age of 6, which is actually longer than many SMA patients live, and that saved his life, likely, the disease has left him profoundly disabled. He is unable to lift himself up from his bed, so he's always horizontal. He's unable to breathe by himself, so you can see there's a tube that comes out of his throat to allow him to breathe. He's also unable to eat unassisted. So this disease has really ravaged his body. His younger brother, Javier, who's there in the middle on the bike, has had a very different course, and that's because his older brother was born with this disease. Javier was actually diagnosed in utero, so it is before birth. And that meant that as soon as he was born, within 2 weeks of birth, he was given the same treatment as Matteo, but much earlier. And although he is still affected by the disease, he can walk with some assistance, and he's able, as you can see, to ride a bike. So he's had a very different course. But the real miracle in this photo is their younger sister, Amelia, who you can see all the way over on the right-hand side. I think in this photo, taken not long ago, she's about 7 years old here. Now, Amelia was also diagnosed in utero, so before birth, but the treatment she received shortly after birth was a brand new therapy, a gene therapy, that was injected where a virus containing a functional copy of the gene that's broken in her genome was injected into her spine. That virus traveled throughout her brain and passed that gene into many of the cells throughout her brain and spinal cord, and replaced the bad copy of the gene, or supplemented the bad copy of the gene with a functioning copy of that gene. And as a result, seen here at 7 years old, Amelia is a functionally completely healthy little girl with absolutely no trace of the disease. So the reason I'm showing you this family is partly because it illustrates, I think, beautifully the power of genomic medicine. That is the ability to identify disease or predict disease before symptoms actually begin to appear, and then to identify exactly the right molecular intervention to prevent that disease from occurring in that particular patient. So that I think is really important. But the other thing this photo shows is just how fast this field has moved. And that will be a theme of my presentation tonight, is the fact that we are living through an unprecedented transformation of medicine, largely driven by a set of technologies that are converging in this field of genomic medicine. So the three areas of technology that I thought I would talk about tonight are, firstly, of course, genomics, and I'm going to spend most of my time discussing that, which is a set of technologies that allows us to look at the cells in your body and the DNA and other molecules within your body at unprecedented resolution. Secondly, I'm going to talk about data science, which of course is super familiar to most of the people here in this room. But data science in this context is another set of very rapidly evolving technologies that make it possible for us to think about making sense of all of the data that's now rolling out from genomics and use that to make predictions about the risk of disease, for instance. And then finally, I'll also cover therapeutics, which is, again, an entire field of technologies that are converging increasingly on an ability to actually use biology itself to correct or treat many different diseases. So let me start with genomics. To think about genomics, and here I'm mostly going to talk about genomics in the context of DNA sequencing. So most of the presentation I'll give today is about DNA. I thought I'd start by giving those of you who don't have a background in DNA or genetics a really lightning overview of what DNA is and what it does. So the most important thing to know about DNA is, well, firstly, it's a physical molecule. It's found in every cell in your body. But it is also it's an information carrying molecule, and it encodes that information in a string of these 4 letters, A, C, G, and T, and there are lots of them. This is a random chunk of the human genome that just whizzed past. At peak speed there, that was moving past at around about 50 letters per second. If you were to read all of the 3 billion letters in one copy of your genome at that speed, 50 letters per second, it would take you about 2 years to zip through the entire genome. So that gives you a sense of just how much information can be compressed within one copy of your genome. If you were to read both copies of your genome, the copy you inherited from your mum and from your dad, it would take you about 4 years. I don't recommend doing this, but that gives you a sense, I think, of the scale that we're talking about here of information. And that information is packed up and coiled in a really efficient way inside every single cell in your body, with a handful of exceptions like red blood cells that don't have DNA, but almost every cell in your body has the same DNA sequence that's packed up inside these 23 chromosomes that are passed down as each of the cells divides to create you. Now, your DNA, of course, famously is unique. It is, in fact, even more unique than a fingerprint. there has never been another human before who has had your DNA sequence, and there will never be, again, another human who has your DNA sequence. And in fact, that's true even if you're one of a pair of identical twins, like these cute guys here. Identical twins are not, in fact, identical at the genetic level, because although they are born from a single egg and sperm, during the first few cell divisions that result in them then cleaving out to become two separate embryos, there are always a handful of mutations that occur that result in there being a very small number of genetic differences between identical twins, often not enough to change their physical appearance, but enough that if you sequence their genomes, you could actually tell them apart. And your DNA is not everything. It's not destiny, as I'll come back to later, but it has a huge influence on many different aspects of your body. If you look at the person next to you, you and that person differ from each other at about 1 in every 1,000 positions in the genome. So that's 3 to 5 million genetic differences. A lot of those differences between you and your neighbor don't have any impact at all. They're found in bits of the genome that don't actually do anything. But others of those differences will influence the way that you look. They have an influence on the way that you behave. They definitely change your risk of a whole variety of different diseases. And much of the wonderful diversity of humanity that we see around us is a consequence both of environmental variation to some degree, but also of substantial genetic variation. Now, your DNA is also, importantly, a history book. It records in its sequence the history, not just of your immediate family, but actually your deep ancestors. And in fact, by looking at the DNA sequence of humans and a whole variety of other species, it's possible to reconstruct a tree that allows us to see the patterns of relationship between every species that exists today. This is one version of this tree. This is now a little bit out of date, but there's lots and lots of species that are drawn around this tree here. And for those of you who are really curious or have very good eyesight, humans are in this insignificant little branch up the top here. And most of this tree, of course, is composed of tiny unicellular organisms that we don't generally think about. But the beauty of DNA is that because we're able to align sequences against each other and see the relatedness, we can actually reconstruct this tree with a remarkably tight degree of precision. So that's your deep history, but we can also use DNA to understand more about your more recent history. This I think is a really neat example. It's now a pretty old paper. This is from back in 2008, but this was from a study led by John Novembre. What this study did is to take an unusual set of individuals. These are individuals born in Europe where all 4 of their grandparents were born in the same country. And they took, so each of those people, they're different countries, but for each person, all 4 grandparents came from the same country. They took those people and they looked at their DNA, about 1 million points of variation across their DNA, and then they clustered those individuals using principal component analysis, basically a way of looking at similarities between things. They clustered people purely on the basis of their genetic similarity. So in this plot here, there's no actual information about the country that these people's grandparents came from. This is purely how similar, the closeness, proximity to 2 people here is purely related to their genetic similarity to each other. And if you then look at the colors over on the right-hand side of the countries that these people come from, you can see that there's a remarkably good concordance between this genetic map of Europe over on the left and this physical geographical map over on the right. And of course, that corresponds to the fact that for a fair chunk of European history, or at least recent history, people have tended to stay in more or less the same place. And so as a result, they tend to be slightly more genetically similar to each other than they are to other people. This is not perfect. You can see lots of cases where there are people who are intermediate between clusters or who fall into different clusters, but there is information, real information that is carried in your DNA that corresponds to the postcodes of your ancestors. Just to give you a sense about some of the information you can get, I thought I'd take a little bit of a tangent and talk about my own genome. This is pretty old data that was generated by a company called 23andMe. Those of you who follow the news will know that 23andMe is on the cusp of bankruptcy right now, and we don't really know quite what's going to happen to my DNA and the DNA of tens of millions of other people who've been genotyped by this company. But what 23andMe did was to give you a chance to generate genetic information and then provide some really interesting analyses about the information that's present in your own genome. I could spend a whole presentation talking about various things that I've learned, none of which are particularly health relevant about my DNA. I thought I'd just tell a couple of stories from it. The first is one of the things you can do with 23andMe data is compare yourself with other people who've also been studied using the same approach. And you can look to see how similar you are across different parts of your genome. Now, in these three plots, each of the horizontal lines corresponds to a chromosome. So chromosome 1's at the top, and there's the X and Y chromosomes down the bottom, for instance, lots of others in between. The colors correspond to how similar I am to another person that was also studied by 23andMe. Light blue means I am 50% similar to that person, and dark blue means I'm 100% similar to that person. That is, both copies of my DNA from my mum and my dad are identical to the two copies that exist for that person. Now, most of you will have, some of you, I think, will have guessed that the person on the left-hand side is a parent. Indeed, you are correct. That's my dad. So I was very relieved to see that I am, in fact, 50% related to my dad at every position in the genome, apart from the X chromosome, as expected. So he is, in fact, my dad. That was reassuring for all of us. The person in the middle here is my brother. Now, because of the way that genetic assortment works during reproduction, my brother and I differ from each other. So in some places we are 0% identical, some places 50%, some places 100%. It turns out my brother and I are slightly more closely related to each other than either of us are to our dad, which is mathematically possible, but kind of cool. And that brought us closer together, I think, as a SIB pair. So that was great to see. And the plot on the right-hand side, reassuringly, is my wife. And it turns out I'm not related to my wife in any way whatsoever. And that was, again, great for both of us. So that's one piece of information we can get. We can also look again at ancestry, at the geographic history of our ancestors. And on this plot, which kind of resembles a worldwide version of the principal component analysis map that I showed for Europe earlier, I am this green plot right up in the top left-hand corner, and I cluster boringly, as expected, with the European cluster, mostly sitting with people of British ancestry, exactly as expected, given the oral history of my family. But there was a surprise, and that was that although the vast majority of my genome is unambiguously and pretty boringly British, and that's all the blue bits here, there were two chunks shown here in orange that were very, very clearly of South Asian ancestry. These particular patterns of variation just are not seen generally in people with exclusively European ancestry. That was a puzzle. My brother, it turns out, also had some of the same chunks of South Asian ancestry, and in fact, he had a couple of other bits of South Asian ancestry and other chromosomes as well. And all of those bits came from my dad. My mum has no trace of South Asian ancestry at all. So it's interesting. We had no oral history at all in my family of any non-European ancestors. So it took a fair bit of digging into genealogical records to figure out where this actually came from. And it turns out it is this distinguished man who has a beard that I can only ever aspire to grow. This is Captain Abel Henry Chapman. He's a British soldier, and he served in India for about 15 years in the mid-1800s. We don't know a huge amount about what he did in India, but we do know that he came home with a wife, Mary Fraser. Mary's parentage also is a little bit fuzzy. We know that her dad was British and traveled to India. Her mum is unknown, and she was born in Calcutta. And the genetics lines up with Mary Fraser being 50% Indian. And so those little pieces of DNA that I can see in my genome and my brother's and my dad's genome almost certainly descend from Mary Fraser, born of mixed ancestry in Calcutta, who then came home with Abel Henry Chapman, who then apparently just pretended that there was no Indian ancestry. And so as a result, there was just no oral trace of this particular ancestry in my family. So that was a cool discovery that came out from that work. Now, that was a diversion, and I want to jump back into what the other sort of more useful things that we can learn from DNA. Before I do, I just wanted to say one of the analogies that gets used a lot with DNA is that it's a blueprint for life. And I think that's actually pretty misleading. This is not a rigid or deterministic plan for your body. It is, I think, better thought of as a kind of recipe. That is a set of instructions, an incredibly complex set of instructions, but a set of instructions that carry meaning, but are then interpreted in the context of your environment and a whole variety of other influences, in the same way that a recipe varies enormously depending on exactly what ingredients are used and exactly who prepares it. You can end up with very different outcomes. And that's also true. This is an unflattering analogy, obviously, but this is also true for humans and for this DNA recipe that we carry. This is a pair of identical twins, so born with nearly identical genomes, a few differences, as I said earlier. Are born in England, and the lady on the left stayed in England. She never smoked, and that didn't see much sun because, of course, she lived in England. The lady on the right, in contrast, moved to Florida and took up smoking for most of her life. And you can see very clearly that despite having almost identical recipes, these two women ended up with quite different appearances as a result of the environmental influences that they experienced. And so I think that's a really important reminder of the fact that DNA is important, but in most cases, it's not destiny, with the exception of some of these very severe genetic diseases like spinal muscular atrophy that I showed earlier, where basically 100% of the people who carry the genetic causes of those diseases will go on to suffer from it. So we now know a huge amount, thanks to, I'm not going to go into the technologies that we're using here, but thanks to a whole variety of approaches that have been used to understand which bits of DNA correspond to which aspects of our appearance or behavior or disease risk. We now know a lot of these, in fact, hundreds of thousands of positions in the genome are now very clearly statistically correlated with a whole variety of different outcomes. These include obesity, where we know actually many thousands of parts of the genome associated with obesity. We know bits of the genome associated with your response to drugs, both in terms of the efficacy of those drugs for you, as well as your risk of side effects. There's also a lot known about the genetics of appearance, so we can predict actually with almost perfect precision your eye color and your hair color from your DNA, as well as a whole bunch of other traits. So actually, here's an interesting one. If I said to you guys, if I said to you guys that some of you here in this room will know with absolute certainty that you've eaten asparagus the next time you do a wee, how many people know what I'm talking about? A few of you, right? The rest of you may have no idea about this, but it is the case that there are some people in the population, I'm one of these people, where your genetics actually results in you metabolizing asparagus in a particular way. And also there's another set of genetics that can determine the way that you receive chemicals that are processed from that asparagus. As a result, for those of us here blessed with this particular genetic trait, when we urinate, anywhere within about an hour to 6 hours after eating asparagus, there is a very distinct smell. That means you can absolutely tell if you've eaten even a relatively small amount of asparagus. And those of you who are looking at me like I'm crazy, trust me, this is absolutely true. Talk to the people who have their hands up and they will convince you that that's the case. But this is absolutely genetic, and it's one of many, many traits that we now understand how they happen, not just at a genetic level, but now actually increasingly at a molecular level. So all of the parts of this molecular machine that lead to the process, for instance, of increased obesity. And of course, that's also true now for severe disease. We know, like that spinal muscular atrophy story, we now know many thousands of genes that are associated with very severe genetic conditions, such that when there's a mutation or a genetic change that breaks that gene, it results in these very severe diseases. And my team has been spending a lot of time, both in my previous job in the US and also here in Australia, using genomic technologies to understand those causes of disease. Now, I'm not going to talk about the specific technologies we're using here. What you mostly need to know is that there are a whole bunch of really fancy machines, and I'm talking really fancy. They have lights that flash. They vary enormously in size. Some of these are enormous, big machines that can sequence DNA. The one on the bottom right is very cool. This is a machine developed by a company called Oxford Nanopore that is actually just about the size of a thumb drive, and you can still use that to sequence a person's entire genome, which is incredibly cool. So it can actually sit on a bench top. You can put DNA in, and it will actually give you sequence data that is very readily usable. So that can now be used in the field to do things like pathogen testing, for instance. Now, the speed of change in genome sequencing has been blisteringly fast by the standards of any technology. In fact, faster in many parts of the curve, even than Moore's law for the pace of change in computing. So to give you a sense of how fast it's moved, the first human genome was announced, we announced its completion back in 2003. It had cost us at that time it was well over a decade of work by a global consortium, and it cost the world somewhere, the number is not exactly clear, but somewhere probably between $2 and $3 billion. So a huge amount of investment in sequencing that first human genome. When my team sequences a genome now, we can do it in less than a week, and it costs us well under $1,000. So that is a many, many orders of magnitude decrease in cost and increase in speed over the course of that time. To give you a sense of how fast that is, this is a Ferrari Enzo. It's a lovely car. If you would have bought a Ferrari Enzo back in 2003, it would have cost you about $650,000. Now, if the pace of change in automobile manufacturing had been the same as what we've seen in genome sequencing, this same wonderful vehicle today would cost you less than 10 cents, and it would accelerate about 1,000 times faster than this one did. So as you can tell, genome sequencing has moved incredibly quickly, definitely much faster than the manufacturing of cars. And as a result of that precipitous decline in the cost of sequencing and the fact that genome sequencing is increasingly valuable for predicting your health, we now know well over 50 million people around the world have been sequenced. This number is now pretty out of date. It's almost certainly over 100 million now. And much of that sequencing is now being done in the context of not just research projects at scale, but actually increasingly in the context of healthcare. And that is because it is now routine. For instance, if you have a cancer, in many cases, one of the frontline tests for that cancer will be to take DNA sample from the cancer itself, as well as from your blood, to identify the specific genetic changes that are responsible for that cancer forming, and also to identify the drugs that are most likely to kill your tumor without killing you. And if you have a child or a grandchild who's affected by severe genetic disease, like the spinal muscular atrophy family I showed earlier, again, the frontline test now is to sequence either the entire genome of your family, or in some cases the exome, which is the particular pieces of the genome that correspond to protein sequencing bits, and then use that to identify the specific genetic changes that have actually caused that disease. And again, this is something my team now does and helps support in a very routine basis. And these two fields are the leading edges of genomic medicine. But genomic medicine is already touching almost every aspect of healthcare. And over the next decade, I believe there won't be really almost any part of medicine that isn't perfected in a fairly profound way by the availability of cheap and accurate genomic information. So the ability to generate information at that scale is really what's driven my career. And just to give you a sense of how that's worked and how that corresponds to the pace of change in genomics, when I did my PhD up in Sydney back in 2003 to 2007, again, we only had that one human genome sequence available at that time. And I spent my entire PhD studying a single gene, in fact, a specific single variation in that one gene. In 2007, I moved to the UK, to the Wellcome Trust Sanger Institute, to do my postdoc position. And while I was there, the first wave of a project called the 1,000 Genomes Project had just started. I was able to work with hundreds of human genomes, and I studied every gene. And in particular, was really interested in looking at genetic variation that breaks the function of particular genes. And it turns out there were lots of these variations in everyone in the population. And then in 2008, when I moved to the Broad Institute of MIT in the other Cambridge in Massachusetts, we started to get to the point where we had first tens and then eventually hundreds of thousands of genome sequences available. And there we were able to start thinking about studying all of the patterns of genetic variation that existed across the population. And the reason I was really interested in pulling together data from many, many tens of thousands of people was that as my lab started to sequence the genomes of individuals affected by rare genetic diseases, it became clear that to make sense of that data, we needed to be able to compare the DNA sequence of patients with the DNA sequences of hundreds of thousands of other people, so that we could understand for each of the changes we found in the patient, how common is that in the general population? And if it's really common, you can exclude it because it can't possibly be the cause of a rare disease. And then we can just focus in on the few hundred variants that might actually be responsible for that patient's disease. And that resulted in the Genome Aggregation Database, which is, or GNOMAD, a project my team spent most of their time working on during the time that I was there in Boston. To give you a very brief overview of what we did with GNOMAD, this project now includes sequencing data from over 800,000 people. It's a huge amount of data. It's over 20 petabytes of raw data for those of you who are computationally focused. It's a lot. It was donated to us by over 100 research studies. So we aggregated together data from lots and lots of different consortia. These were mostly researchers who were sequencing tens of thousands of cases and controls for particular common complex disorders, things like type 2 diabetes, for instance, or heart disease. We processed all of that data through the same pipeline. This was extremely difficult. It required working on the cloud and building a whole new set of algorithms to actually be able to operate at this type of scale. And then we called genetic variants across all of these samples simultaneously, so that we had, at every position in the genome, we could actually say that we had seen the same variant in the same way in each of those different individuals. And as a result, across now all of the 800,000 people that we sequenced, some with genomes and some with exomes, so there's just, again, that roughly 2% of the genome that's protein coding, we've now identified over 1 billion variants, 1 billion positions in the genome that are variable within the population. So that means at about a third of all of the positions in the genome in general, we have seen at least one person in the population who has a genetic change at that particular site, which is kind of incredible. As this starts to increase by the next order of magnitude that we go up as we move into the tens of millions, we will begin to approach the point where almost every genetic variant that is compatible with life will be seen in at least one individual in that data set. And so we can really use that to study a lot about the impact of variation on human traits. And importantly, what we did with this cohort was to make all of the genetic changes that we identified freely available to the world, so that anyone who was working on looking at DNA sequence data from a patient with rare disease could go to our website, nomad.broadinstitute.org, look up any genetic change that they found in their patient and see how common it was in the 800,000 individuals in this data set. For those of you who are interested in genetics, there's a lot of information packed into this website, and it's been used now a lot by both clinical clinicians and researchers, about 75 million page views or so far. And that is mostly because it is the largest and most freely accessible database of genetic variation now in the world. So that has been it's been wonderful to see how often that's been used. So that's all I'm going to say about genomics, and I'm going to move pretty rapidly through. I promise you I'm not going to go in as much detail in data science and therapeutics, but I did want to give you a little bit of a tour through these areas because they are so important for genomic medicine. So in the context of data science, many people in this audience know more about data science probably than I do. But what you can say with data science is that, like what we see for genomics, almost doesn't matter what you plot, you see a curve that looks like this. This could be cloud computing capacity, could be the amount of data that's now being thrown to train new large language models, for instance. We have continued to see an exponential increase in our ability to handle and make sense of very large amounts of data. I'm just going to give you one example that I think illustrates this really nicely, which is the problem of predicting the structure of a protein from its sequence. Now, again, for those of you without a biology background, proteins are the doing molecules in the cell, mostly. So most of the things that your body does are actually being done by proteins. They form the structures within your cells. They are the enzymes, for instance, that change one type of chemical into another type of chemical. And the reason they're able to do that is because they form these exquisite three-dimensional structures. They go from a long string of amino acids, individual chemicals, and then they collapse up and fold up into this very intricate three-dimensional structure, and that is what allows them to do their thing. In this particular case, this is a protein called CFTR. This is a protein that's famous because mutations in this gene, in the gene that encodes this protein, cause a terrible disease called cystic fibrosis. And this protein's function is that it actually spans the membrane. So there's this bit of the protein up here, which actually sits in the membrane of a whole variety of cells in your body, and it passes sodium through that channel. Ions through that channel, and that is really important for balancing the amount of electrolytes within and outside your cell. And as a result, when you lose or have this protein is damaged, that results in a change in salt balance that badly affects the lungs, resulting in the most severe effects of cystic fibrosis as well as other tissues in the body. Now, the protein folding problem, that is predicting, given a string of amino acids, what three-dimensional structure it will fold into, is a problem that is both incredibly important because we can't develop new therapies targeting these proteins unless we know what structure they form, but also incredibly difficult. And in fact, for decades, people have been throwing a whole variety of different types of algorithm at this particular prediction problem. So how do we go from a string to a three-dimensional fold? These are the results from a competition that's been run every 2 years, here showing the starting date back in 1994, where a set of protein structures that have never been seen by the world, firstly, the sequence of those structures is shared with a whole community of predictors. Each of those predictors then does their best guess at what the protein structure looks like, and then there's a rigorous assessment to determine how well they do. The y-axis doesn't really matter here. All you need to know is that it is a measure of how good those predictions are. And you can see here there was a pretty steady increase, roughly, in accuracy of these prediction methods from 1994 for probably a decade or so, and then a rough stagnation. So about a decade where it was actually very difficult to see any material advances in the accuracy of protein prediction. In fact, in some cases, it looks like we went backwards for a couple of years. Now, so things were stagnating pretty well until 2018, when there was suddenly a jump, and that was caused by the release of a brand new algorithm called AlphaFold, which had been developed by Google DeepMind. It's very clever, I guess, AI, machine learning algorithm that had been trained specifically on a huge variety of protein structures that were available in the public domain. It used a very clever machine learning approach to do that. And as you can see, it demolished all other competitors and represented a really substantial jump in accuracy. And then DeepMind did it again in the next competition 2 years later with another, in fact, even larger jump in prediction accuracy. And what's amazing about the accuracy of AlphaFold predictions is that they now rival the level of accuracy that we see for many experimental methods for solving proteins. So we now have, although the protein folding problem is not solved, there are still large swathes of the protein space where AlphaFold does not actually do a great job of making predictions. But for many, many proteins across the genome, we now have pretty good structures that we can use for all types of things. For instance, in the context of that map I showed you earlier of CFTR, and here this was provided by Claire Bycroft from DeepMind, we can actually color this using the patterns of variation that exist within this gene by looking at the Nomad dataset. And what we can see here is that these red versions of the protein are the regions that are depleted for variation in the general population. The blue regions are the ones where there's actually quite a lot of variation seen in the general population. And it turns out those red bits are the really, really important bits, where if you get a mutation in one of those regions, it is most likely to cause disease. So AlphaFold plus Nomad is able to now do a really neat job of actually predicting which of the new mutations that we find in a patient might actually be responsible for their condition. So that's all I'm going to say about data science. I did want to touch now very briefly on therapeutics. And again, I think motivated by the story of that family that I showed earlier. So historically in therapeutics, this has been we need therapies to be able to treat these conditions. We have many, so well over 95% of all genetic disorders have no effective therapy whatsoever right now. But over the last 5 to 10 years in particular, we have seen a wave of new therapies that are developed using biology itself to treat disease. These include things like, for instance, RNA therapeutics, which allow us to turn particular genes on or off within a cell. That's the same type of therapy that was used to so very rapidly develop these effective COVID vaccines as well. We also have gene therapies that can take an individual affected by a severe disease and insert a new functioning copy of that gene into the cells that are affected by that condition. And that was the type of approach that was so effective in Amelia, the little girl that I showed on my first slide, is this gene replacement approach. And then we also now have approaches called gene editing using an enzyme called Cas9, that's often called CRISPR technology, and that can actually very specifically go in and target particular changes in the genome, and in some cases revert those back to the original form. So this now provides new therapies for things like sickle cell disease, for instance, where we can take cells out of a person affected by this disease, edit that particular mutation back to the original version, then put those cells back in to now provide functioning copies of a patient's own cells that can help to alleviate the symptoms of that disease. Now, all of these types of therapy would have genuinely sounded like science fiction if you were to talk about them 20 years ago. Even 10 years ago, in many cases, gene editing would have seemed crazy. Every single one of these types of therapies is now being used actively in patients, at least in the context of clinical trials. And it now looks like over the next decade, there's a good chance that we will start to be developing really effective therapies for many of the remaining rare genetic disorders that don't yet have things like gene therapy like that spinal muscular atrophy. So the next decade looks like it will be a genuine golden age for therapeutics, so long as we can get things right when it comes to aspects like the regulatory challenges, as well as, of course, as the challenges of reimbursement. So that is this family, I think, really beautifully illustrates the ways in which we can use genomics, data science, and therapeutics to identify disease and then to develop approaches that allow us to intervene. And I think over the next decade, this will result both in an increase in our ability to predict someone's future risk of disease, to diagnose disease that's already occurring in those individuals, as well as to develop and then deploy the most effective therapies to treat that particular disease. So I think this is an incredibly exciting time to be working in the field of biomedicine as a result of the convergence of these therapies. But I did want to finish with a challenge, and a challenge that my team now thinks about a lot, which is the challenge of equity. And that is, in Australia, of course, we are an incredibly diverse country. More than a quarter of us were born overseas. More than half of us have at least one parent born overseas. And that is just as true of the patients around Australia who are affected by rare genetic disorders, of course, as well. And one of the challenges that creates is that in these big resources of genetic variation, like the Nomad projects that my group worked on, they don't actually capture all of the different types of variation that exist in the Australian population. And in fact, we know for sure that there's at least 5 million Australians who belong to ancestry groups that are either completely missing or at least almost entirely absent from existing resources like Nomad, or in fact any existing global resource of genetic variation. And that matters because it means they are less likely to get an accurate genetic diagnosis, and they're more likely to get an uncertain result when they are subjected to genome sequencing. They're also less likely to be able to access precision therapies. So solving this equity challenge has been a big focus of my team's work, the Centre for Population Genomics. We're based jointly at the Garvan Institute of Medical Research in Sydney and the Murdoch Children's Research Institute just up the road here in Melbourne. And we really have three focus areas we work on. The first of those is partnering with underrepresented communities around Australia to build a much more representative data set of genetic variation. And that's through a deep engagement with these communities that has gone on over the last couple of years. Second big program area for us is to bring together information on genetic variation from projects like our genetic diversity map, and then combine that with other types of information like information about which genes are switched on or off within individual cells in the blood to understand the impact of variation on biology. And then finally, a big focus on understanding the genetic causes of rare genetic diseases with an eye towards eventually improving our prediction of risk, particularly in these underrepresented communities, as well as identifying new targets for therapeutics. I'm just going to mention two programs very briefly today. The first of those is the Our DNA program. We are currently in phase 1 of this program. The goal is to really address this critical gap in the representation of many Australian communities in existing databases. And to do that, we are recruiting 10,000 individuals from 10 underrepresented communities. We will, by the end of this year, reach just over 900 individuals, initially from Filipino and Vietnamese ancestry. And over the coming years, we'll be working with communities of Indigenous ancestry, as well as those from Oceania, more from Southeast Asia, from the Middle East, as well as East Africa as well, all living here in Australia. For each of these individuals, we're collecting and generating genome sequencing data, as well as collecting live cells that we can use for a variety of downstream biological experiments, and health information that allow us to link genetic variants to both biology and health. And then we'll be making the summary data from these data sets openly available in most cases, although not for all communities, such as Indigenous communities, for instance, to empower as much as possible the diagnosis of disease in patients from those particular backgrounds. And so that project is very much underway right now, and we hope to have reached that 10,000 goal within about 2 years. The second project I just briefly wanted to mention is using data science here to improve the way that we can diagnose patients like Amelia affected by severe genetic disorders. And this is a new funding that we got just this year, at $8 million from the Medical Research Futures Fund, to build a new national consortium to apply new analytical technologies, particularly AI tools, to improve the diagnosis of rare disease. This is the Australian Alliance for the Safe Use of Genomics and AI in Rare Disease, or ASGARD consortium. And this will be developing, testing, and applying these tools at scale to improve diagnosis, starting with over 6,000 individuals from rare disease families for whom my team has already collected together genomic and clinical information, and all of that now stored in a secure cloud-based repository for analysis. So my final slide, I just wanted to mention what we need in order to be able to make sure that genomic medicine benefits Australians, but particularly benefits all Australians. The first of that, and the Our DNA program is really just a start here, is that we need to be working in a very responsible way to build up a collection of genomic and health information from millions of diverse Australians. And that's really the only way that we can ensure that we have the information we need to provide predictions, diagnoses, and therapies for Australians of all backgrounds. Secondly, and critically, we need to be investing in building a pipeline of world-class data science talent. Biomedicine is increasingly, again, I think many people here in this room are very well aware, biomedicine is increasingly becoming a data science. Well over half of the people in my team at the centre, for instance, are people who actively write code. And as a result, we need to ensure that we are well placed to bring people into biology who can actually make sense of all of the data that's now flowing out from these genomic resources. And then finally, we need to ensure that the tools that we build for prediction, diagnosis, and treatment of disease are fundamentally equitable. And that means not just thinking about equity at the high level, but actually ensuring that we deeply involve all of the communities that live here in Australia in the process of building our research resources, as well as designing the processes for delivering accurate care back to individual communities. And if we can get that right, then I think we have a very bright future ahead of us over the next decade of genomic medicine. So with that, I'll finish by saying a huge thanks to everyone for listening to me over the last little while, and I'd be very happy to take any questions. Before I do, I just wanted to mention, if you're interested in working in this broad space of genomic medicine, feel free to check out the Population Genomics website or drop me an email. We're always looking for excited young people interested in applying particularly data science to the context of healthcare problems. And otherwise, thanks very much for listening.
Thank you, Daniel, for this exciting and great talk. Do we have any questions from the floor?
Thank you for the very nice talk. Given your 23andMe example and what's happening to the company, I was wondering, when you go out for data collection to the underrepresented communities, what kind of conversations do you have regarding privacy and ownership of their genomic information?
A lot. And this is a great question, and one we thought about a huge amount. In fact, one of the reasons that the RDNA program is really kicking off in earnest this year, as opposed to when the centre was launched back in 2020, is that we have spent a solid 2 years of that period very focused on community engagement and understanding both what is the levels of literacy around genetic topics across these communities, but also what do they want and what do they need to be able to see from a project to feel comfortable participating in it. So those conversations included a lot of questions around security and privacy. A big question that came up and was genuinely very difficult to answer up until this year is, for instance, what would happen? What are the implications for me from an insurance perspective? So if you were to find something interesting in my genome, what would I have to disclose that to an insurance company? What would that mean for my premiums? And very fortunately, this year we saw a concerted campaign that us and others were involved in. It was actually led out of the Monash here in Melbourne, to create legislation that would prevent insurance companies from actually using genetic information to discriminate against people. And although that's not legislation yet, we've now got a strong commitment from the government to actually get that out, which is fantastic. But there's lots of other questions that people have, for instance, around security and privacy. We've built our entire systems with that in mind. So for instance, one of the things we do is all of the information we store is cloud-based, but we actually store the identifying information entirely separately from the genetic data. And in fact, for anyone in my group, they only get temporary access to be able to link those two things together for a defined period of time for a defined set of samples. And so that basically means even if an intruder was able to get access to the authorization of a particular individual, they couldn't systematically link all of that data together. And then there's also a lot of work we've done around encryption and constant penetration testing and other approaches to keep that data secure. We can never make a guarantee of privacy and security, but I think we're at a point where we are well ahead, at least, of the vast majority of research projects in Australia. There's a bunch of other questions that came up. One common theme is around forensic use, for instance. So if police come knocking on your door and say, give me access to your database because I want to figure out if someone in there is related to a murderer, a DNA sample that I just found at a murder scene, would you have to fork over the data? And we've been able to work with lawyers and with our institutions to come up with a clear answer to that, which is that if the police come knocking and just ask randomly to trawl the database to look for someone and they have no particular reason to expect that that person or their relative is in the database, we do not have to give access. But of course, if they come back with a court order, which generally would require very specific information, we will hand that over under very defined circumstances. So yeah, it's been a lot of conversations, but I think we're now at a point where we can actually give the right assurances to communities to participate.
Hi, thank you again for the talk. It was really well done. I just wanted to ask specifically about the access to genomic data, and specifically your opinion on who should be able to read and interpret genetic data that comes back. Say you go to one of these private companies, and I think it's become more increasingly prevalent that these companies will give you information on level of disease risks that you have, and it goes straight to you as a person, as your information. It can be kind of vague to interpret that genetic information and the level of risk, and people can start to panic without clinical advice. And I want to ask your opinion on how the interpretation and the access to data should be controlled or to avoid panic, or the data should be displayed in a specific way to give greater information?
Again, a good question, and one that was particularly discussed a lot as the first wave of personal genomics companies like 23andMe first came out. The question there was, should any genetic information ever be provided back directly to a consumer without there being a clinician in the loop? And this was a very heated debate, because I think there was a clinical community that was very concerned about the potential harm of people learning information that might scare them. On the other side, we had people, and I would count myself in that group, who saw that view as often very paternalistic and were really irritated at the idea that we would need an intermediary between us and the data that we wanted to look at. There's been a lot of studies that have been done since then about how people respond to genetic information, and those have been mostly extremely reassuring. So it actually turns out if you give someone information about genetic disease risk, even if their alarm transiently spikes, it will actually almost always return back to a baseline level. And that's true actually even for pretty devastating findings of genetic information, is that people generally don't panic when they receive that information. They tend to take a while to process it, and then their anxiety levels go back to normal. So that's quite reassuring. But at the same time, there's also been a lot of research into how you can present that information in a way that is more accessible to individuals and makes it easier for them to actually make sense of it. And I don't think there is any one answer in this situation. I think there are some people who would greatly prefer to receive their genetic information through a clinician, and that should absolutely be their right. They should have the ability to access that information in the way that they want. At the same time, I believe if someone wants to get access to their own DNA, and they would prefer to do it with no intermediary involved, they should absolutely be allowed to do that, and that should be their prerogative to be able to take that on. Other people disagree with me about this in general, but I think it should be down to individual choice as to whether, how you look at your own body and the information that's in there.
Thank you.
Thank you for the amazing talk. I wanted to ask you, what is the level of de-identification of data that you think is necessary? With the advancements in machine learning, we have lots of questions about bias that arises, especially if it's like male, female, like sample ratio indifference or things like that. So we also need to protect their privacy with data security measures. So we also have to de-identify the data for research purposes. So what do you think, or have you, in your experience, is the level of that you've experienced?
Another great question. And here, of course, the concept of de-identification with genetic data is slightly absurd, because I spent a fair bit of time early on explaining your DNA is absolutely unique. So if I have a DNA sample from you and someone else has a DNA sample that they know belongs to you, they will absolutely be able to tell that that is your DNA sample. At the same time, the risk of re-identification varies enormously on the particular circumstance. So in general, we feel pretty comfortable sharing individual level genomic information, firstly in a context where there's clear informed consent, where people have thought through the various risks involved and they agree to share data. Secondly, where there is a well-defined research purpose, and in the case of our DNA, that also requires that the proposed research be unambiguously of benefit to the community. So there needs to be a sort of a community benefit component to that as well. In those situations, although there is some risk of re-identification, in order for someone outside to re-identify that particular DNA sample, they would already need to have DNA information from that particular individual. So they would need to have that person's DNA sample and then use that to match the data that's in our database. And in general, under those circumstances, it's actually unlikely they would learn anything particularly new. It's likely they already have a fair bit of information about that person that they're looking to explore. So our sense here, and the way that we've talked to participants about this, is that there is a material risk of re-identification that occurs in any genetic study. It's never zero. At the same time, in most of the studies that we're doing, the probability of that resulting in harm is actually pretty low, but participants should go in with their eyes open and make sure that they're aware of that risk.
Thanks, Daniel. So my question regards the gap in that you have in rare inherited disease diagnosis and large structural variation. So I wanted just for you to talk about some of the challenges and limitations in aggregating large structural variation, and how do you see that impacting that rare disease diagnosis gap that we currently see.
And did you say aggregating structural variation?
Yeah.
Yeah, good question. So I didn't talk about structural variation in the talk, but for those of you who don't know the term, this is basically a particular type of genetic variation that involves a really large rearrangement of DNA. So for instance, you could have a piece of DNA that's deleted or duplicated or inserted or switched around. These types of variations are surprisingly common in the population. They also cause a significant burden of rare disease as well. So I think what you're pointing out here is the fact that for structural variants we have substantially more limited information in resources like GNOMAD about the patterns of these variation across the population as a whole. That's fair. And we've done some work in this space. So we have GNOMAD SV, for instance, where we have now about structural variant information for about 75,000 whole genomes, but again, not representative of the entire population, and only using short read next generation sequencing data where it's certainly not perfect capture of all of the different classes of structural variant. I think in order to really have a perfect sense of what the SV landscape looks like in the population, we need to be using long read sequencing, and we need to be doing that at population scale. So long read sequencing is a class of variation, a class of sequencing technology that, as the name implies, looks at very long strings of DNA, so you can actually stitch these things together. And that's coming. So we've got 10,000 genomes currently being generated in the US on a diverse cohort from the All of Us program using, in that case, Pacific Biosciences long read data. And there's a variety of other projects around the world currently working on generating that long read sequencing data at a pretty large scale. At the same time, we've got groups including, we have a collaboration with Ira Jefferson's team at Garvin, and there's other groups looking at this around the world, applying long read sequencing to look at patient genomes as well. And I think that is the path forward towards a complete ascertainment of structural variation within individual patient genomes, but then also having the ability to look up the frequency of that very specific rearrangement in the context of really large cohorts like GNOMAD, is to have long read GNOMAD resources available.
Thank you.
And happy to chat to you. I may not have answered your question there, but happy to chat after the talk if that's useful.
Thanks, Daniel, for the informative talk. I have a couple of questions, and I would like to know about the view on traditional medicine versus genomic medicine. Because for the traditional medicine, people have a lot of confidence in that, right? For genomic medicine, still lots of people don't really kind of believe that can provide a treatment or healing for them, right? So what are your views? How can we bring, that confidence to the public? And because as it's getting, and technology is advancing and lots of work is being done, we need a lot of skills and resources for this, right?
Yeah.
How can we, bring people to study it, gain those skills and resources, all about confidence? What are your views on that?
This is something we've thought a lot about, not quite in the setting of healthcare, but definitely in the context of recruitment for the RDNA program, where we feel that in order for consent to be truly informed, we need to be talking to people who have at least a passing understanding of the concepts of genetics and genomics. And so, but also one of the side effects we hope of having the RDNA program is that it is an opportunity for us and our team to get out into communities. We spent a huge amount of time this year, for instance, as we've recruited individuals of Filipino and Vietnamese ancestry, going along to cultural festivals, having stalls, talking about genetics and genomics and the benefits of genomic medicine. And then at those events, we also recruit individuals who are interested into the program itself to donate their blood samples. I mean, the things I've seen from that process is that firstly, there is, entirely unsurprisingly, there's a very low level of understanding of genetic information out there in all communities. And this isn't just in these underrepresented groups. This is broadly population-wide. That means I think the scientific community as well as the clinical community have a lot of work ahead of us to ensure that we are actually actively working around the process of informing the community, sharing resources that are actually accessible, not just talking to other scientists, but also talking to the communities about the benefits of the technologies that we're working on, the risks and the challenges, and how these things will actually impact their lives. So this involves, there's a lot of work to be done here. One of the things we have been working on, in fact, we now have a set of resources providing information about genetics and genomics that's now translated into 10 languages, one of the 10 most commonly non-English spoken languages in Australia. That's freely available on our website. We'll be working over the next a couple of years to translate that into as many different languages as possible so that those resources are accessible. And that's partly as an opportunity for recruitment, but I think also an education component as well. But none of this is going to be easy, and it will actually require lots of education, but also, unfortunately, often pushing back against a heavy amount of dis and misinformation that exists out there. And we have seen, of course, over the last few weeks, a series of people being proposed for positions, very senior positions in science and health in the US, who would actually be very aggressive in providing incorrect information at very large scale. So I think our work is going to be cut out for us in pushing back against that type of misinformation.
We're gonna close this public lecture today. Thank you all for participating and listening to Daniel, and thanks Daniel for this amazing talk.
Thanks everyone, much appreciated.